my lab currently has developed a technology with a very high accuracy where we can tell where mutant DNA is located. And mutant DNA is very important because that's a really specific marker of malignant cells. We don't have very good, proteins tend to be rather poor markers of malignancy because proteins are not so different between malignant and benign cells. But if you can pinpoint the mutations, then you have a way of finding where are the malignant cells, how are they infiltrating, are they approaching the line where the surgeon cut, so meaning that maybe there are malignant cells also on the other side, or are we clear of that? And how has the mutations evolved over time? You start with a handful of mutations and tumors mutate all the time, so over time they will accumulate more and more, so you can see the clonal history of a tumor spatially distributed. So DNA can be interesting also. RNA, of course, is even more interesting because RNA gives you the identity of the cells. And that's already well underway. There are lots of technologies to sequence RNA, but the faster you can read them out with single technology or similar, the better. Protein now is yet another area, and I've been arguing that the proximity ligation has advantages over other protein detection techniques because we can improve the specificity and we can look at more complex features such as interactions, modifications, and so forth of the protein. But on top of that, of course, as we just discussed, anything else that you are interested in and where there are enough different events that you want to distinguish typically can be encoded as DNA. That's done more and more now. We have the epigenome, so we want to know also which parts of the genome are accessible for the transcription machinery, which ones, even if they're not currently being transcribed, which ones are ready to be transcribed. That's what you learn at the level of the epigenome. You study the nucleosomes and the technology called ATAC-seq tells you which parts are not so hid from view, but they are active and can be accessed by the transcription machinery. So these are just some examples where we want to use DNA readout to learn more about biology.